Good morning or good afternoon, depending on which side of the world you are. My name is Zoltan Perstegi. I'm the Vice President of Product Development at RotaChrom North America, and I'll be your host for today. Let me start by saying a couple of words about RotaChrom because we have some new guests. This is the second of our webinar series. Before we start, so RotaChrom is the inventor and only producer in the world of automated industrial scale centrifugal partition chromatography instruments. Established in 2014 in Europe, RotaChrom has presence in North America, Asia, and Europe with applications in the pharmaceutical, natural extracts, food and beverages, and cosmetics and fragrances industries. Today's webinar will feature Andras Gaspar, RotaChrom's product and patent manager and scientific advisor, an esteemed scientist with research and work experience from Australia to Germany, who is currently based in Hungary, but working with our customers all around the world. I'll give you a couple of housekeeping notes, and then I'll hand over straight to Andras. Your cameras are off and microphones are muted throughout this session, but you'll find a Q&A button at the bottom of your screen. So if you have any questions, add them there. And we set aside ample time during today's webinar to answer as many of these as we can during or at the end of the session. And with that, let's head over to Hungary.
Thank you, Zoltan, very much for the nice introduction. And I would like to also welcome you all to this second webinar of ours and the first one in 2021. So with that, I would like to also give you a wish from our side and give you wish you a happy new year. So as Zoltan already introduced, for today's agenda, we would like to discuss and give you a brief insight to the industrial scale partition chromatography. Centrifugal partition chromatography is somewhat unknown in the separation world, so that's why it would be advantageous, I guess, for a lot of people to give you a short insight. How does that work? Where is this positioned in this purification space? Then, of course, regarding to the purification and regarding the complete process flow in the hemp and cannabis industry, we also go into detail and go into some discussions on the investment side and what are the considered factors that you need to look out when you're going to establish your process. And of course, via some case studies and some listing, we're going to detail of our applications and then try to emphasize how versatile this approach is. And at this stage, I would like to also draw your attention for an upcoming webinar, which is going to base basically a consecutive part of this presentation on a benchmark comparison platform that we created over the time, which will probably give you a really nice insight on this landscape. So without further ado, let's just start. And since partition chromatography is somewhat unknown even for the chromatographic world. Even myself, I majored in analytical chemistry and had a PhD in analytical chemistry. First time that I heard of partition chromatography was at RotaChrom. But in order to put that in a storyline that is somewhat really not really something new, I would like to share you some of the historical background and how this whole technology evolved. So around 1940, Richard Singh actually laid the foundation for this technology with the technical background and the theoretical background as well, and created that time so-called countercurrent distribution. And even in that decade, Lyman Craig already came up with a really nifty apparatus, an instrument that was able to separate, purify compounds. But that sort of instrument was a kind of really complicated glassware. But that sort of instrument was not that time. The major drawback was that it's really labor intense and was not able to automate it. So it took a couple of decades to figure out this, and a Japanese scientist and inventor, Yoshihiro Ito, came up with an idea, and actually he introduced a gravitational force into this whole game. And with this planetary centrifuge that he designed, and that was basically second most important milestone in this whole evolution. And that stage, of course, since this gravitational force was already introduced into this automated system, several companies started to work on the upscaling, because purification, it's not really something that is on a benchtop happening, but it's rather like on industrial scale. So first, a Japanese company started to work on a stacked disk concept, and unfortunately that ran into some technical barriers which did not allow that time to increase the volume and the throughput. And around in the late 90s, early 2000, several companies, especially in France, started to work and started to rethink that stack design concept and move away from that to radial cell design. And in that stage actually, this was somewhat at the dead end. So those companies, with their design, they understood that what's the design lacking. They created a different design, and that unfortunately did not really deliver that breakthrough that they were hoping for, really scaling up the partition chromatograph. So, and that period, actually, RotaChrom has also started to work on its prototype design, and they came up with this patented design around 2004, which was completely different from the competitors, and that actually enabled the company to really establish a product line which is on a true industrial scale from the purification perspective. And just since it's something we should be really proud of, it's with a Hungarian heritage. And just in order to give you a fun fact, so to speak, Hungarian invention in the purification and in the separation science is not something unheard of. If you just think about Mr. Horváth, who actually built the very first HPLC that we know today, or we're talking about Mr. Molnar, who actually laid the groundation work for the reverse phase HPLC, which is most probably the most common separation technique around the globe. My colleague last year in December had the first seminar, went into great detail to explain the technique, the theoretical background, and how does the partition chromatography works. So for this reason and for sake of our time, I didn't really want to go in details. I just simply collected a couple of key notes that probably for those who are not familiar with chromatography or even partition chromatography might be useful. So this type of separation technique utilizes two solvent systems that are immiscible, and these solvent systems are actually the crucial and the core part of the separation. And as I mentioned before, that one of the key feature of that partition chromatography is that we're using a gravitational force, and in this case, the gravitational force is created by a rotation. And the way how the separation, the mass transfer occurs is that a mobile phase percolated through the immobilized stationary phase, which is another type of liquid in this rotor. And the major difference that is going to give us relative advantage over traditional liquid chromatography with solid stationary phase, that lies in a partition. And in order to deliver that resolution that purification is all about, you want to resolve analytes from each other, many or just some, or analytes from impurities. That's basically, it's done by the selectivity. In case of liquid chromatography, you are able to increase and enhance your selectivity in a way that with that you were able to increase your resolution, and you are able to detach analytes from a matrix, and you are able to fraction them. While on the other hand, the traditional HPLC's answer for resolution is basically a kinetic optimization, which is usually it's related to capex burden solid stationary phase optimization. So what are those advantages that are deriving from this selectivity optimization? As I told before, that it's a stationary less chromatography. So with that, you have almost an infinite combination of liquids that can create this separation environment. The method development, unlike any other chromatographic method, doesn't really require instrumentation at the beginning. It's just a normal shake flask method where you create and you analyzing the partition coefficient of those analytes within your solvents, and this basically this result that you receive there and selectivity that you created with your solvent system is one-on-one scalable. So whatever you find on your benchtop, it's going to be almost linearly scalable to our system. It's a forgiving technology, which means that since there is no stationary phase, there is no tedious method development. There is even on the run, so it's on the production level. If something goes wrong, if the pH, if the salt content or whatever it's for some reason it doesn't really work, or your method during the method development, something happening, there's nothing irreversible. So this means that you just simply throw away and regenerate the solvent or the sample that you use, and you start over. There's no solid stationary phase that is going to be ruined, and for a couple of thousands of dollars you need to reinvest in those. Since there is no stationary phase in this system, there is no absorption, so everything goes in, comes out. Like any other chromatographic method, this has also a nice way to add another dimension to your separation. So you are working in a batch mode, you going in a separation, you focusing on some analytes, and then you collect those analytes and you reintroduce that analytes and using a different method. So you give a certain orthogonality to your system. That's without question. And unlike other techniques that are on the market, that like harmonic distillation, where there is some chemical alteration to your input material, here in this chromatographic method, there is nothing such, no such thing occurs. And since there is an infinite combination, and basically the swapping the solvent system is the way how you operate. Your operation is extremely flexible. You can change complete separation platform within your shift, or whatever commercially makes sense, or within a day, within hours, within minutes, you can change those things. So it's extremely useful if you have multiple revenue streams, and then you want to optimize those outputs based on the customer, your customer's requirement. So how RotaChrom actually evolved in this hemp industry? As I told you, around mid 2010s, RotaChrom came up with and commercialized partition chromatographs. But these are basically core instruments, and then core instruments was developed for pharmaceutical industries, where solvent regeneration is something that is not really common. And as we entered this hemp space, it turned out very quickly that the customers were not able to handle and not prepared for the volume and for the solvent handling. So we came up with a production system that's already taking care for you all that's solvent, also for the impurity line. Collects the solvent, concentrates the solvents, and regenerate the solvent and feeding back into your system. That was at that time even we pushed a step further, where we said that it's not only the impurity line, so the waste fractions needs to be addressed, but also the compound of interest line, especially if you're considering the fact that solvent residue on your system is always a question mark. You don't really want to provide products that at the end comes back to you because it may have some solvent residue. So that's we addressed over the period that we are acting in this industry as well. So we are able to provide you. I presume at this stage we are the only provider on the purification side who is able to provide you an integrated solvent and compound of interest regeneration system with the core purification system. So that's, of course, that comes with several advantages. That flexibility still remains. So whenever you swapping to another solvent system, in that case you are also able to modify and alter the complete regeneration system. In that case, it's something that the recovery suddenly working for a different solvent system without any issue. Of course, the major benefit for investment considerations that your OPEX costs, so mainly your consumables, that the main cost contributing factor is significantly reduced. And of course, it's not only on the consumable side, but since it's a fully automated system, the attendance, the personal attendance is also limited. So basically on the OPEX cost, that labor costs included, that's also reduced. It's also, we came to an agreement and formed a strategic alliance with Heidolf, a premium rotavapor manufacturing company who tailor-made a system for us as well. So with that combination, we are able to deliver product to the market and to your customers that is safe and providing a reliable finishing touch your value stream. So since this discussion is or this presentation is about investment considerations, and the first and foremost important question is that where do we locate ourselves or instrument in this purification process or the complete process chain? Well, we rather at the really end of that process chain. And when you are considering investment and cost burden of that, of course, it's always important to address key questions. And this is what during customer interactions, this is what we always do. We also designing together and try to iterate together with the customer to be actually not only our purification step, but we also considering the complete process flow. So it's a kind of process engineering at the same time. So what are these key components that needs to be addressed in order to have a really optimized and cost optimized solution? So you have to know what are the cost contributing factors of the different processing steps. We have here a really normal, quite straightforward application, but nowadays you have more and more applications where winterization and filtration is done by nanofiltration. So it's something that needs to be kept in mind. What is my easiest and the most flexible process that I want to use? What are the volume requirements? What is that I'm able to process? What sorts of level I like to process, because that's really going to influence the setup. Is it something that for 100 kilo a shift, 1,000 kilo or tons of material, green material that needs to be processed? And most probably all of you know that this sorts of industry that we are in, it's really volatile. It's evolving extremely fast, and the target is keep moving and changing. So that sorts of process flow needs to be not extremely tailor-made, but you should keep it as flexible as possible. So the questions that one day I'm going to do isolation, next day I'm going to do remediation, the third day I'm going for minors, or I'm going for some different matrix, some fermentation broth instead of natural extract. So all of these things are actually question mark and needs to be kept in mind that to be possible to react fast, you need to stay flexible. You need to have also understand and address what are the value chain, what are the product lines that I want to bring on the market, because for each different products, purification step will be different. What are the requirements on your market? Is there any regulatory requirements, API standards? Is there any customer relevant standards? And again, it's just like I mentioned before, your operation at this stage at that volatile environment needs to be as flexible as possible. So just to give you also an idea how the individual steps are impacting your investment, we did several analyses with several customers on the individual steps, and as you see here, without giving you too much details, that's going to be on our consecutive presentations in the future months. But at this stage, this is something how it's basically look like, what that cost and capex burdens are extremely uneven, and we can say that independently of the purification solution, most process cost and process burden, and might be also capex burden coming on the purification. So the more you optimize your process, the less capex burden you're looking at. So we thought that it might be easier and better to present those via case studies. So those design considerations and factors. So of course, the main target for any type of investment or business start, that you want to have the highest added value product as possible, because you want to have the highest margin and profit as possible. And of course, you want to lower your capex. You want to have it for free, of course, but you want to have the lowest capex as possible. So we brought you today 4 different case studies with 4 different from 4 different customer discussions. The first one where we actually, by simply optimizing the output material and the properties of that output material, the final finish, that higher added value suddenly appears. The second one is rather something that coming today really more and more popular, is waste valorization. We have several customers where the volume, the sheer volume of that input is just simply doesn't make sense to process in a known way. So we need to swap and change the process in a nifty way. Then of course you can even top up that one. We have several clients and where the discussion is really something where the input material is already a nice finished material, nice distillate, but the throughput requirement is extreme. So you need to have also a solution to be ready to reduce as much as possible your capex. And it's not only always about investment that has an impact on your total cost of ownership, but also that the purification method, how a method is well set, it's crucial. So the first one is the customer approached us that they were not really aware of the fact that a full spectrum crude contains several other unwanted components that may reduce your final value on the market, such as chlorophyll, flavonoids, terpenes, and all of those things that usually does not give the nicest touch on your final product. So, and of course, since the crude extract needs to be THC remediated, we did that as well. But then we also suggested to the customer that what would happen if we just simply move and insert a couple of steps in between, where we might able to polish further that initial crude extract. And although these things has capex burden, at the end of the day, because of those capex burden are uneven over the entire process, we were able still reducing that capex, because the purification capex was lowered due to the volume reduction, and the customer received a much higher added value at the end. The second option was there where the volume was much higher. So we faced with around 400 kilo of extracted material that needs to be processed per shift. And that time they were already aware of the fact that that material needs to go through the entire process. So winterization, decarb, distillation. But what we offered them and recommended them that we should introduce a step where we suddenly creating crystallization via crystallization mother liquor that considered to be in a lot of cases just like industrial wastes. Unfortunately, because the THC content is even higher, so it's like hazardous waste. And many of the clients that we have, these sorts of material is lying there and they are not able to process it. So the idea comes from that section where we have that mother liquor, which is basically the residue of the crystallization. And in that specific case where the CBD content is extremely reduced, all those minors are still there, and then also the THC is there. So with that, we were not only able reducing the necessary equipment for the purification, because of course here in that step where after the crystallization, you significantly reducing the input volume on the purification step, but you were also able to create several revenue streams of this customer. And the customer was able to have the terpenes and decided if they want to mix it for the flavor's sake, or if you just want to sell it differently. And it's the same valid for the CBD crystals, if you want to sell it as is, or you want to increase the concentration of your broad spectrum oil at the end. And of course, I was already mentioning orthogonality. We offer them also a combination with a smaller instrument. In that case, you were able to provide another value stream where you fractionate and then separated the minors out of that. And we all know that probably minors today is the hot topic and highest seller on the market. Then this was also kind of a solution for a customer that moving in the beverage industry. So that volume, that more over a ton of fine distillate had to be processed and remediated. So we also recommended the similar waste valorization stream that significantly over, as far as I can recall, as I remember, it was around 60% reduction on the capex burden for the purification process. And of course, at the end of the day, the customer realized that although each step has some certain loss, but the final product and the capex and the combination of those things was really a way to go for. And finally, when it's nothing to do with investment, it's rather like working as an expert on the solution. A customer of us approached with a legacy method from our competition. So basically the competition left them with a really weird method that was set up for them. And then they approached us that if we can do something about their throughput and if we can optimize their revenue. So this is what we did. Our experts came in, studied the problem and the task, and modifying the method that was inherited and changing completely to our method, they were able to reach a much higher load capability, and they were able to improve their revenue stream extremely. So, and of course, if you are into invest, if you are into considering what would be the best and most flexible solution for my problem, then of course it's always advantageous to look for the appropriate purification solution. In-house, we also did this homework. We called it the benchmark, but we call this, of course, the technology selection support. We created this tool and monitored basically the major relevant technologies and companies that are acting on this market and able to provide you a purification solution. This is since this is our space where we're moving at, so it would be advantageous also for us to understand. And we're looking into solutions that are on the industrial scale, and of course they are present on that in this industry. So it's not that they have an existing industry, and then they thought that would be nice to also act in the cannabis space. But some providers that they are really after, they develop method, they provide you with white papers and support. And of course, we also compared and shortlisted those advantages and limitations of the different technologies. And the key partners, key parameters, KPIs that we were, where we were actually monitoring a lot and compared a lot, KPIs, but the key parameters that we were actually checking is, of course, the quality, the throughput, the load capability, what sorts of consumables and human resources, so labor requirement, labor intensive applications, and of course, what are the capex burden to those different technologies. And just to for the sake again of this presentation time and keep it in a frame, time frame, we're not going to go into details, too much details in this presentation, but we like to draw your attention that the next presentation of ours is going to focusing on this selection support tool and going into much further detail on the relevant technologies and the competitive landscape. So as we mentioned earlier before, that we are RotaChrom is on this market from the beginning, and over this period of couple of years, we've been in contact and with customers, and we installed several of these instruments all over the globe, and specifically in the US, where actually, and as I already mentioned also, that this industry is keep evolving and keep changing in an excessive speed. So that's why over this couple of years, we developed and helped to our customers with well over 50 different methods, and the list is just keep growing. So at the really beginning, the CBD isolation was something that was quite common. Then THC removal, THC isolation, and the minor isolation is at this stage. Pesticide remediation is also a huge question mark at this stage. So it's just give you an idea that that is not something that we just pop in and install the instrument and leave you as is, but we also provide you with a great support, and we have an established background in this hemp space. So I like to sum up this presentation. So the bottom line of that partition chromatography and what RotaChrom delivers with that industrial scale solution stands for flexibility. It's an extreme flexible design and process, which is you need it definitely in this space. It's scalable, so whatever you have and through the process optimization or the instrument product line that we have, it's relatively easy and simple to scale your production. It's efficient since it's many of those technical and consumable burdens that other legacy chromatography has, this does not have, and it's versatile. So as I mentioned, that we developed several methods for several applications, and these are things that it's at your disposal. It's the lowest possible costs, delivers the lowest possible costs, OPEX costs for that flexibility that is provided parallel. And it's providing really a wide array of targets, solutions for those targets. Since we, from the beginning, acted as almost like a process optimizer and a process designer, not only for the purification step, but also for almost the entire production process, we do believe that we could be also your trusted advisor. And again, it's I hope that I was able to demonstrate that if you are into this business, if you are into setting up a business, you really need to optimize your process chain. So with all that, I'm finished. I'm finishing this presentation. I hope that you find it interesting and you have some now some insights in this hemp space and the problems that or the solutions that you may have on the on the investment side. And then also like to draw your attention to the upcoming next session, where we're going to go over this provider landscape and give you a detail of our findings. And with that, I turn back to Zoltán, and thank you very much for your attention, and we are open for questions.
Thank you, Andras, for the very insightful presentation. While I look through the questions, I would also like to draw your attention to our upcoming webinar, which will be held by Andras once again. So he will be talking about comparing and contrasting centrifugal partition chromatography with legacy chromatography. I think that's a very exciting topic, especially for those who are considering purchasing a purification system and platform. I think this will surely be very exciting, and I'm sure that we will see even more numbers and insights than today. So we're going a little deeper even than today in today's session. So save your seat now and ensure you won't miss this opportunity. You see the registration address, so that's bit.ly/RotaChrom webinar, the same URL that you used for registration to today's session. And now let's see some questions. There is a more technical question, so I will ask Andras to answer this one. Is there a partition coefficient range at lab flask scale that is considered suitable, and how does it relate to throughput or resolution?
Yeah, that's like any type of liquid-liquid extraction or liquid-liquid distribution. There is a sweet spot to every type of analytes. We usually keep those sweet spots of the distribution of the partition coefficient. It's really related to that analyte that you are after. So this is something that we need to discuss presumably offline, because it needs to be calculated and properly adjusted for your analyte. But there is always a sweet spot that enables you to have certain retention. If that coefficient is too small, then suddenly your product analyte is just coming in the really beginning. So, or if it's just too high, it will stick forever on that column. So there is always a sweet spot that can be found during this shake flask analysis.
Thank you, Andras. Moving on, next question is about the square footage used for the system. So I will actually jump in and answer this question to our viewer. Actually, there are, as you've seen, the complete system consists of 2 different rooms. The C1D2 room is roughly 20 by 40 feet, while we have a separate room for the rotary evaporators, which is roughly 20 by 20. But it's really dependent on there are multiple downstream and upstream processes, so this will be dependent, but the rough square footage requirements for the system is 20 by 40 and 20 by 20, I would say. Moving on to the next question, how's the maintenance costs? There's a question about that. Well, what I can say is that all of our systems come with a 1-year full warranty. So that's the first point to know. And all of our systems are installed on site by our technical team, and then there's always method development time included as well. So this is how RotaChrom systems are installed. We're not going to leave you with a piece of hardware that you don't know how to use. It will be properly installed, and you will know how to purify your specific compound. So we will develop a base method basically for all of our customers. When it comes to after the 1-year period of maintenance, well, we offer a full line of parts that are needed for maintenance, and we also offer maintenance packages for our customers. I usually compare or contrast this to something like Apple Care. So you can purchase this package, and then all of your maintenance needs are included If you're interested in more details, yep, I would recommend that we speak directly at sales@rotachrom.com. You can reach out to us, or just use the phone number that you see on screen there, and we can give you further details. One other question to, and I think this is going to be to Andras. So you used the term orthogonality a couple of times. Can you please explain what you mean under that?
Yeah, I'm keep getting this from the sales guys that that is not the nicest word to continuously use. But since I'm coming from analytical chemistry, and I used to have this on many occasions, it's if I want to simplify it and really translate it to normal day language, you have the option and the possibility to giving an additional dimension to your separation. So once your separation starts, you inject your material, you run it whatever runtime you have, and then you're going to fractionate those. So basically slice up your separation. But if you have those slices, if you have those fractions, and then you run those fractions via another method that is completely different from the first one, so you're going to have another dimension to another separation, which you can slice even further if you want. And this gives you also the potential that you can really focus on analytes, target materials that may coelute, that may come together, or they superimpose on each other. So at the first step, and you are not able to focus on it, or they coelute. And then if you have this second stage with a completely different method on the very same instrument, that needs to be emphasized, then suddenly you are able to have a much higher resolution. And this option, as I mentioned earlier, the number of options or number of combination of solvents that can be used is just infinite. So that's basically orthogonality means, adding another dimension to your purification steps.
Thank you very much, Andras. And that actually concludes our session for today. So thank you once again for your time and attention. I hope you found our session useful. Don't forget that we have launched our podcast. It's called Purified. It's available on our all major podcast platforms, where we always dive a little bit deeper into the topics of our webinar. So Andras will be featured on our second episode. If you want to sign up for that, you can go to Spotify and just search for Purified by RotaChrom, and you will find our podcast. So subscribe and don't miss the deep dive into today's session. Our second webinar is concluded with that, but I hope to see you again on February 11th. With an even deeper dive into the comparison of the different chromatography technologies and platforms available. So don't forget to register. Take care, and goodbye for now.
Thank you very much. Bye. Bye, you all.